Thanks for joining us today at Singleron's inaugural Single Cell Innovator webinar. As Khalil mentioned, I'm Jonathan Skolnick. I'm the General Manager here at Singleron Biotechnologies in Singapore. And today I have the pleasure of opening our four-part Innovator series by introducing one of our founders, Professor Rong Fan, who's really a true innovator in single-cell analysis and in particular in its applications in immuno-oncology. So Dr. Fan is a professor at Yale, where he holds dual professorships in biomedical engineering and pathology. And I think today we're going to see how those connect to each other. And his work has earned him numerous awards and fellowships, including the NSF Career Award and the Packard Fellowship for Science and Engineering, amongst many others. And today we're going to have the opportunity to hear Professor Fan talk about single-cell multi-omics applications in cancer research and cell therapy. So Dr. Fan, thank you for joining us this morning for you, and the Zoom room is yours.
Yeah, thank you. Thank you, Jonathan, for the very kind introduction. And thank you, everyone, for joining the seminar and from everywhere around the world. And good morning, good afternoon, good evening. And so I'm going to go ahead and kick off my talk today, focusing on kind of single cell multi-omics technology for the study of kind of cancer immunobiology, or how to better develop and how to better characterize immunotherapy products. And so broadly speaking, my passion over the past 50 years in the immuno-oncology space. So this is my disclaimer. And so I would like to start my story with this kind of news I read back to 2007 when I was a postdoc at Caltech. And so that time, okay, now you probably noticed I was reading a lot of stuff, not just the scientific research articles back to that time, because I had literally no training in biology during my PhD. And actually those news articles are nice for me to understand kind of at very high level why those kind of biological questions are important. In this case, what got my attention is the HIV vaccine trial conducted by Merck, and the vaccine turned out to be very, very potent. At first, a T cell vaccine, but after phase 1, 2, 3, completely failed in phase 3, and people got together to discuss why. Okay, this article published in JEM is sort of like a conference expert opinion piece to discuss why this highly promising trial failed and what we should do next. And so, of course, I think human and monkey different. You need to better understand human biology. But one thing that got my attention is the assay they used to characterize sort of the anticipated immune response induced by the vaccination is kind of single parameter gamma interferon production. It turns out that's still widely used in the vaccine industry nowadays, called gamma interferon ELISpA. But immunobiologists back to that time already show you that's not enough, all right? Even for just one type of T cells, like a helper T cells, and you need to look at many different effector molecules, not just the gamma interferon, and ability for one T cell to produce many different immune effector molecules really dictate their overall quality, their durability, and potency in no matter vaccine-induced immune response or many other kind of immune effector function in different contexts, including cancer. But then I realized, okay, you need a tool to characterize not just the gamma interferon production, but many other immune effector molecules, those different proteins, and how many effector proteins can be produced by T cells. And I got a list from Mara Rodder, who was leading the immune characterization division at a vaccine research at NIH. He gave me a list about 40 to 45 different proteins, and okay, can we measure all those? But 2006, I call that an ice age of single cell omics. One of my best friend, I think Bo Huang, he was a graduate student with Dick Zera at Stanford. and he got just a one cell in the complex microfluidic device, and you lyse the cell counted just one or two types of protein. Wow, that's a science paper, all right. And so analytical chemists back to that time were thinking how we can do like a electrophoresis in a microchannel, such that even the amount of materials as small as just from one cell, you can still kind of quantify the amount of protein from just a one cell, but you can look at just one or two types. I then realized, okay, that would not work for the specific case I discussed in the Merck clinical trial, because you need to look at hundreds, thousands, or even tens of thousands single cells to really understand the population scale heterogeneity. But I also need to look at, as Mario told me, like up to 40, 45 different proteins. And that time that was kind of like a science fiction. I didn't realize that science fiction because I knew very little about biology or the biomedical technology. And so, oh, seems like that's the tool we need. Let's go ahead and develop. And so that's what I came up with when I was postdoc with Jim Heath. And so instead of uh, sort of separating the proteins using gel electrophoresis or capillary electrophoresis, I said, okay, I learned something called a protein microarray, but very different protein microarray developed in Professor Jim Heath's lab called a DO, DNA encoded antibody library. You can detect a larger number of proteins because each spot detects a different protein. So you basically can kind of avoid this spectral overlap challenge when you use multi-color fluorescence detection to look at different proteins. Now you don't care because now you use a spatially encoded spots to detect the different proteins. All right, so that's not something okay people feel so striking about nowadays because there's a microarray technologies what was commercialized, I think, for detecting the transcripts and the gene expression. That's a DNA microarray, and also commercialized for protein detection. But you would not think that works for single cell protein detection. But I thought actually it's possible. If you miniaturize the device such that each microchamber you used to encapture a single cell is as small as 1 nanoliter. So you do your math. If a one cell in 1 nanoliter, that's equivalent to 1 million cell in 1 ml. All right, when we culture cells in a petri dish in a flask, that's very typical cell culture concentration or density. So that means if this little guy is producing the same amount of protein in this tiny nanoliter microchamber versus in the flask, it should have more or less the same protein concentration. So the question, can we put all those different DNA antibody spots, or in this case, stripes, into nanoliter microchamber? So that worked out, but that's not the focus of my talk today. But just to give you a little bit of flavor, how the single cell analysis started, at least in my own journey, but afterwards, I had the privilege to work with many experts at the UCLA, in particular the pioneers in the immuno-oncology space, for example, Professor Tony Rivas. And what he told me back to that time was, okay, immunotherapy was kind of like you're throwing the glass jars from the top of UCLA medical buildings, and most of them going to break. But occasionally the glass jar stay okay, kind of intact, and you just have to continue to do that because some patients may benefit. It is largely due to the sort of the pioneers like Tony that really persevere, the belief in the power of immuno-oncology. And now I think one of my cancer center director told me is that now every oncologist is the immuno-oncologist, because that already becomes a cornerstone, one of the cornerstones in entire oncological science or cancer treatment. So when I moved to Yale 2010, I think around that time it's another interesting inflection inflection point in the whole history of single cell biology. And you were able to look at dozens of proteins and to realize high-plex protein measurement, including the technology I just discussed, but other technology came out around that time, such as CyTOF. You can use the metal label to look at dozens of surface markers to better kind of better phenotype or better immunotype different cells in extremely heterogeneous population. So I love my device. Okay, I took this device to Yale and talked to my clinical collaborators. Can we apply this technology to characterization of immune cells in the context of cancer immunology? And I realized, although I love it, but my clinical collaborators had no idea how to use this complicated design microfluidics, and that becomes engineering problem. And since I'm in engineering school, I said, okay, let's go ahead and take on that challenge and further develop the technology to make it much more scalable. You can so measure hundreds or even thousands of single cells, and each cell. Okay, we didn't realize that we didn't demonstrate the full spectrum immune effector proteins for all T cells yet before I started Yale. And so, okay, we still need to meet that goal told by Mario Rudder, like 42, 45 different immune proteins. And also we can kind of really redesign the whole device to make it much more manufacturable, and potentially that can be kind of commercialized, kind of industrialization can further scale and impact. That's my goal. So a number of publications to show you how the technology evolve, but also our own interest in immunobiology beyond T cells. We demonstrate how that works for the characterization of functional heterogeneity of myeloid cells. And so, for example, this is the first demonstration of this for the two-plex immune effector protein analysis at a single cell level. And so how the technology works now, that's a much simpler device. And if you just dispense the cells into those microtroughs, and they are encapsulated in the microtroughs, you put the glass slide on top, and you have the microarray in the kind of running vertical as the different stripes, and each cell in a chamber producing whatever effector molecules. You can capture them by those different stripes, and then you read out the different stripes using immunofluorescence, using the DNA microarray scanner, and you know what proteins are produced by those individual cells. Then we look into the macrophage innate immune cell heterogeneity, but also how to kind of communicate, because what we're looking at now is the protein secreted by individual cells to mediate their peripheral signaling. Now you can look into this cell-cell interaction, how to working together to shape their collective response. That's a very interesting sort of system biology, protein signaling research we published together with my colleague, Professor Catherine Miller-Jensen. And it turns out not just the kind of normal immune cells, and cancer cells also produce a lot of effector molecules, cytokine, chemokines, growth factors to mess up the tumor microenvironment, as showing here. And so on a normal hematopoietic cells cluster nicely in those like blue dots, the different distinct clusters, but in a leukemic microenvironment of the bone marrow in this mouse model, so you see the effector function or functional profile really kind of spread over the whole map. That produce a lot of many of those cells, for example, here produce tons of effector molecules are not supposed to produce, right? So that's kind of consistent with the hypothesis early on for good T cells. They are polyfunctional, but in this case, the polyfunctional cells are bad. They are causing kind of completely inflamed, messed up tumor microenvironment. So, but anyhow, the take-home message is you need a tool to characterize at a single cell level, and as informative as possible. You can look at tens or hundreds of relevant proteins in this case, and then you don't fly blind. You can kind of really explore what happened for every single cell, and the heterogeneity at a functional level can tell you the mechanism, number one. Number two, that gives you a much better power to develop predictive biomarkers. So how to develop the predictive biomarkers using technology like this? So we got excited about T cells very early on, but if you look at the history of cancer immunotherapy, which is very long history, over 100 years, but I would like to say about like 35 to 40 years ago when Steve Rosenberg infused the IL-2 into the patients, that's because IL-2 expands the T cells in vivo. So that's actually a T cell therapy. And during the past 10, 15 years, you have seen two other major breakthroughs in cancer immunotherapy. One is immune checkpoint inhibitor therapy, PD-1, PD-L1, and the other is engineered T cells, in this case, now TCR, not just the TCR engineered T cells, but also chimeric antigen receptor engineered T cells. And that led to the first FDA approval of engineered cell therapy, but also that how to engineer that was through gene editing, so that becomes the first kind of gene therapy approved by US FDA. So how CAR T cell work to get rid of tumor cells? That's a very, very simple mechanism. One, the chimeric antigen receptor recognize the target molecule on the surface of the tumor cells, like CD19, and that interaction going to trigger the transmembrane signal transduction, and then get into intracellular kinase domain and the signaling domain. And in the second generation CAR, you have two intracellular signal domain. One is this kind of T cell CD3 zeta signaling domain, but you also have the co-activation domain. But afterwards, okay, on the general assumption is the T cell becomes activated, produce porphyrin to poke the hole on the surface of the tumor cell, and then the release gamma interferon, release granzyme B to kill the tumor cell. But the reality is much more complicated. The T cells are producing many different cytokines, but no one knows. And then Kite Pharma took our technology to look into their manufactured CAR T cells. They found the ability for every single CAR T cells before they infuse into the patients, actually before they infuse into the patient, to look at if you do antigen-specific stimulation, how many T cells are polyfunctional, are capable of producing a wide spectrum of cytokines, as defined as polyfunctional strength index. And the patients who could not respond after infusion, most of them have very low polyfunctional CAR T cells. And if you do statistical analysis, that's statistically significant between responder and non-responder. If you are an immunologist, all right, you're going to look at many other things. For example, just like what I talk about, gamma interferon production, and how many of those are CD4 helper cells, how many are like I said, toxic T cells, CD8, and also you can look at T cell differentiation states all the way from kind of naive to central memory, effector memory to effector cells, but none of them can give you a statistically significant difference or association between responder versus non-responder, except this polyfunctional strength index in their cohort. And so now this has been commercialized. I'm not working on that at all in my lab anymore because, yeah, it's already out of my lab. I think widely used in kind of hospitals and pharma and medical centers. But I talk about once you are able to look at tens of proteins, you can understand the biology, you can understand mechanism way better than the single parameter gamma interferon production analysis, right? But can you dream a little bit bigger, even bigger, right? So instead of a panel of immune effector molecules, can you look at the whole genome, whole transcriptome, and all possible genes you are able to detect in individual cells? And then you get really truly unbiased discovery, kind of unlimited and unbiased discovery power to understand the mechanism. So honestly, I think how to isolate the nucleic acids, in particular the messenger RNAs corresponding to gene expression profile, and that was demonstrated for the first time by Steve Quick using this very nice looking microfluidic device, very elegant. And according to my literature search, okay, this paper is actually the first one on the title showing, okay, microfluidic single cell kind of nucleic acid analysis, although they haven't demonstrated can do kind of whole transcriptome-wide gene expression analysis. Why? Because it can now sequence the whole transcriptome, back to 2006. But you can amplify the genome-wide either whole genome or whole transcriptome. That was actually first demonstrated for developed by molecular staging, their lead tech development person, Roger Laskin, developed the technology, this assay called this amplification method called a multiple displacement amplification. The company was located in New Haven and literally just 10 minutes from my office. So I would say arguably New Haven is really the birthplace of single cell genomics. The technology later was kind of acquired, the company was acquired by QIAGEN, and now that becomes the very famous reagent kit called a Ripley G. And our co-founder, my friend, and Fan was actually the R&D associate director and who played very kind of instrumental role in commercializing this MDA product and delivery of this Ripley G single cell whole genome amplification and including the single cell whole genome amplification kit. So as I said, 2006, even though you can extract the RNA from single cells, you can not really sequence them to get an unbiased gene expression profile. But that started to change around the same time as well because NGS came out, right? So 3 different NGS technologies were commercialized, and Professor Fuchou Tang was the first person who demonstrated you can do whole messenger RNA transcriptome sequencing. I believe the NGS methods he used was from Applied Biosystem, but since then, now you can, if you can isolate isolate single cells and extract the messenger RNAs and reverse transcript into DNA and do whole genome amplification using MDA commercialized by QIAGEN through Nanfang. So now you can get a whole transcriptome sequencing at a single cell level. And so what we were working on since then was, I think after I joined Yale, sequencing, that's not my thing, all right? I was not working on any genomic stuff at all, but I had the privilege to collaborate with Professor Sherman Weissman. And so he's really legendary geneticist at Yale. So through that collaboration, we were trying to think, okay, you can amplify whole genome, that you can also amplify whole transcriptome. Can we do both? And I was not very familiar with sequencing business back to that time, but we demonstrated for the first time you can actually co-amplify the whole genome and the whole transcriptome from single cell. And that was done by my talented postdoc, Dr. Lin Han. She's a professor in Shandong University. So that was really the first single cell multi-omics seriously. And a year after, I think 2 papers in Nature Biotech demonstrate back-to-back they call single cell GT6, so genome and transcriptome sequencing. So recently we wrote a piece of review article to summarize the whole history of the single cell multi-omics, and so since then the whole field started to explode. And one of the main reasons the field was kind of booming, exploding, was in 2015 that you are able to realize not just a single cell level, you can do massive parallel single cell analysis of whole transcriptome, potential, I think a whole transcriptome plus epigenome as well. And so the fundamental approach, fundamental mechanism to realize massive parallel single cell gene expression analysis, and not just okay, you can sort of scale the single cell isolation individual wells and a micro titer play. People did that, but the throughput was still rather limited. The fundamental principle is you can barcode individual cells either in droplets or either in microwells. You can barcode all transcripts or something else and from the same cell to give the cell barcodes. And afterwards, you pull on a transcripts or you pull on a cDNA and amplify the sequence. But you can go back to find out whether or not those transcripts are coming from the same cell if they share in the same barcode, right? It's a back-to-back paper in Cell back to 2015, and the two papers share in the same co-author, actually Dave White, because he developed this droplet microfluidics that's truly enabling. So in my lab, we were thinking about the same question, actually, back to 2014, 15. We didn't publish right away, but we were that's good learning experience for me because, as I said, back to 2011, sequencing was not my thing. And so what we came up with, we have this micro wells, we can encapture a single cell and barcode beads and fully kind of cell the bead bead cell pairs. And then we lyse the cell. You can see the lysis is fully encapsulated, fully contained in individual wells, and you get a very good capture efficiency. Okay, so I don't need to go into too much details since a lot of technology came out in the space, single cell multi-omics, but I still think, okay, it's a lot of manual work if you do single cell sequencing. For academic research, that's probably okay. But can we fully automate every single step? And so from this prototype technology to now what has been commercially available at a Singleron, I would like to say the major differentiating point is automation, automation, automation. That's always in our mind how to automate everything from the sample collection to your library and ready for sequencing. We're getting close to there. Some fun fact is probably due in part to our kind of continuous pursuit of the automation of everything in single cell analysis at the I think they partnered with SpaceX and performing the first single cell transcriptional sequencing in the outer space. I think just some fun fact. All right, so back to my passion about CAR T cells. That's kind of the second half of my talk. I want to give you kind of one specific use case of single cell omics, single cell multi-omics, single cell transcriptomics in the study of immuno-oncology. So as I said, we were able to look at a large number of immune effector molecules using our kind of single cell secretion device that made a huge that was a huge leap actually adopted by kind of Kai Pharma and Novartis to better characterize their infusion product. But as I said, it's still not sufficient to understand the mechanism at the fully unbiased and genome scale, and can we really combine the single cell whole transcriptome genome-wide analysis and the protein secretion and the CAR T cell antigen specific activation to understand what's going on at a mechanistic level. So we did that was also a very early exercise that's super fun. But we just grab one type of cell. That's a third generation, turns out, a third generation CAR T cell that has two co-activation domain, not just a 4-1BB, but also CD28 connecting in tandem in a intracellular signaling domain. So we found one thing that's pretty rare, all right? So after we do on the single cell sequencing and also pair the result with the protein secretion, we found you can categorize the cells based on the protein secretion plus the master transcription factor, like a GATA3 and like a T box for type 1, GATA3 for type 2. You can categorize them into the different T cell subsets, Th1, Th2, Th kind of 17 Treg and so on and so forth. But when you look at a single cell level, so it turns out the same cell expressing the Th1 gene also express majority of the Th2 gene. So this is quite a mix. And that made us very puzzled. Okay, what happened? Why those cells are like a hybrid cell state? Because that never happened in our body in normal homeostatic state. But seems like 70% of those cells are highly mixed Th1, Th2 cell response. Number two, we also realize a Th2 type 2 cytokine type 2 function must be important. Otherwise, why you see so many cells in this case, first generation CAR expressing the Th2 function. We would like to do a deeper dive into this question, but also in the context of real clinical study, clinical trials. If you look at even just a CD19 highly responsive patients who are like kind of acute lymphoblastic leukemia patients, the response rate could be as high as 93%. All right, in particular, the patient treated by my friends at a Penn, but after a couple of years, turns out up to like 50% of them relapse. Most of them relapse actually within the first 1 or 2 years. So after they develop relapse, can you still treat them with the CD19 CAR, or you can treat it with something else? You can still do that, but unfortunately, most of the patients die if they develop relapse. So it turns out, I think the CAR T cell therapy is really a major breakthrough in the field, and unbelievable response rate, like over 90%, right? But yeah, so when I first talked to my postdoc, so can we better understand the CAR T cells they manufacture and see can we further improve their therapeutic efficacy? And my postdoc told me, okay, why? Because already 90% of patients responded. Why you need to further improve, or maybe no room to further improve? But if you look at the relapse rate, oh, that's a huge problem. Okay, still a lot more work we need to do. Okay, that's not done yet. So now we try to look into starting from a small cohort, and I think only 5 patients who develop CD19 positive relapse, and 5 patients who are kind of like a longer time flyers, and 2 controls. And in a small cohort, we sequence using our kind of our own microwell microfluid device, and now it's commercialized by Singleron. And so we also add antibodies to do spatial single cell cytotoxic. And so this is what we found. All right. So between the 5 CR and the relapse patients, if you look at all the different cell subsets, okay, we did our very careful annotation. The major subset is a tiny, teeny subset here, the cluster 4, is the only cell subset that can differentiate between the relapse versus the complete remission long flyers. And so that's pretty clear. I think there's cluster 4. And so you see the expression of the cytokines associated with the type 2 immune function, but also you can look at the master transcription factor like GATA3. They are all statistically significant for differentiating between the CR and RL and the relapse. So that's nice. But when you read in the literature, it seems like that's weird because a lot of studies in the past 10, 15 years show you the type 2 or Th2 cells are bad, all right? So the type 2 immunity creates immunosuppressive microenvironment. If you train the patients with PD-1 immunotherapy, PD-1 inhibitor immunotherapy, the patients cannot respond well because they have an immunocompromised or immunosuppressed microenvironment induced by the type 2 cytokines. But in our case, it seems like the type 2 immunity is great. Without that, the patient is going to develop relapse. We thought that's good observation, but 5 versus 5, that's still can we further verify that finding with a much larger cohort? So this is a much larger cohort, 82 patients, and the two different clinical trials. And so now we categorize the patients into kind of five different groups. Okay, long flyers, and they survive over almost 8 years. And because the two clinical trials and some of our patients very likely to be long flyers, but just not yet meet the 8 year bar because they were treated less than 8 years ago, right? And the patients who relapse within a year, within 2 years, and 3 years. So now with all those patient samples and the CAR T cells after we sequence, we got like a 1 million single cell sequence. And after very rigorous filtering, we still have almost like 0.7 million single cells from all those patients. Now we can see all different cell subsets, and including the basal state and the antigen-specific activation state. So now we ask the same question, whether or not those different kind of type 1, type 2 modules and their subsets really differentiate between different patient response groups. Okay, this is how we cluster them together. You can see the presence of all different functional modules associated with T cell functionality, including type 1, type 2, and in particular type 2 and type 1. Again, so now you do see type 1 and type 2, they're somewhat kind of mixed even in this second generation CAR, but still, unlike in the third generation, you can nicely kind of distinguish the Th1 versus Th2, unlike the third generation CAR, they're completely mixed. So now asking the same question in the two different clinical trials, and if you look at on the type 1 scores, and so those cells are high for the type 1 score, so they are potent effector cells. They're going to kill the tumor cells. They produce gamma interferon TF. They also produce granzyme B, and they're the effector molecules to induce anti-tumor reaction. Right. So this is small subset, about like a 5 or 3 to 5% right here. And those cells are producing IL-4, IL-5, IL-13, and also express the master transcription of CALA3. And so if you compare the abundance or the frequency of this cluster of cell across different groups, all right, this is healthy donor controls, the long flyers, and those are the different response, different relapse groups, relapse within a year, 2 years, and 3 years. And you see a major difference really this one. Okay, this cluster 7, the type 2 CAR T cells can nicely differentiate between long flyers and the relapse patients. But if you look at the type 1, in the whole space, people always talk about, can we make the CAR T cells more potent? They produce more gamma interferon, they produce more kind of immune effector molecules to kill tumor cells. But we found actually no major difference within all different response groups. That also explains why the patient initial response rate is so good, like 93% of patients responded, because all the CAR T cells manufactured from at a pen from our collaborators, they're pretty good. Okay, they induce their response right away within weeks. But the problem is whether or not they're gonna maintain a long-term response. It's hard to know. And now we know, and we found that total counterintuitive. It's a type 2 cells or type 2 function really dictates the long-term response. So this is another cohort and very similar results. And you look at the ongoing complete remission patients versus the relapse, you do see statistical difference. So that was further validated using intracellular cytokine flow and also using the technology I described in the beginning, the single cell protein secretion analysis. So that's the gene expression, whole transcriptome plus the protein secretion. But can you further verify that functional kind of type 2 functions also imprinted in epigenetics? For example, in this case, we're looking at single cell ataxic to look for the motifs associated with the type 2 genes, in particular here, the type 2 master transferrin factor GATA3. And you do see statistical difference in terms of GATA3 accessibility in the CAR T cells from the lung flyers versus the patients who develop relapse in the first 1 to 2 years. And so next the question is why? So why you have those type 2 CAR T cells there and the patients can stay remission for a much longer period of time. They are becoming lung flyers. And I always believe the large-scale omics data itself can already teach you the biology. This is a perfect example here to show you. So once you know where those, so within the whole population, which cells are producing type 2 cytokines, but you can also look at within your CAR infusion products, which cells are expressing their receptors. You can do all the ligand receptor interaction to find out the cells that receive that signal, receive the signal produced by those type 2 CAR T cells. And then you do a deeper dive into those cells. And you say, what are those cells? What's the difference of those cells? We found, oh, those cells have significant upregulation of TIM3 and a few other dysfunctional effector, CAR T cell dysfunction characteristic genes. And so, okay, the idea is, okay, what does type 2 subset of CAR T cells do is they regulate the TIM3 positive dysfunctional effector cells to keep them not go further. Okay, turns out even though they are TIM3 positive, they still produce tons of cytokine chemokines. They still do their job reasonably well to fight cancer cells, to kill leukemic cells. But if they are allowed to go further to the full exhaustion, and then you have the whole population shift towards the dysfunction and the exhaustion. You have more and more cells moving down towards that direction. But the type 2 cytokines produced by those type 2 positive CAR T cells really guard the whole population from draining down into that exhaustion state. So that's kind of the mechanism we figure out. And then we can look at that particular population, what's the kind of pathways, biological process. We saw a lot of interesting things, including like the mTOR signaling or the metabolic activities and so on and so forth. And then we also believe the metabolic activities are another kind of co-checkpoint or co-activators you can potentially play with to further improve the effect, the potency and the durability of those CAR T cells post infusion. So a lot more we learn by just looking at the data itself. All right. So furthermore, we were asking, all right, in a cell model, right? So the CAR T cells we manufacture, the type 2 cytokines are important, type 2 function important. Can we kind of modulate those cells by adding those type 2 cytokines to either kind of rejuvenate or somehow make them much higher fitness? And this is kind of in vitro functional experiment. We add interleukin 4 to the CAR T cells in a culture petri dish, and afterwards we sequence. We do see significant kind of increase of so many important gene sets associated with sort of the fitness, overall fitness of the T cells. All right. So what about the post-infusion, how patient responds? I think ideally if you can fish out the CAR T cells post-infusion over many years, but unfortunately those cells are extremely rare after many years. So they probably reside somewhere in the tissue in the bone marrow, but they are very rare. They become like a memory CAR T cells. But we can still look at the patient sera and post-infusion in the first like a couple months, and we found Actually, indeed, the patient after many, many years, they complete remission. Overall, I think their type 2 cytokine level were reasonably higher, okay, compared to the patients who develop relapse. You can see this in the serum samples over the first two months for IL-4, IL-5, and IL-13. So together, seems like indeed, even post-infusion, you do have statistically higher level of type 2 cytokines. And that initial so circulating cytokine levels can to some degree predicted a long-term response many, many years down the road. All right, so there's another way to visualize this data. Okay, we took all the measurements to generate this yield map, and this is not yield map, this is now single cell yield map. All right, so each dot is a patient sample. And so now we kind of group them based on all different cytokines we measure. And then this particular group correspond to the lung flyers. And when you look at what genes, what proteins secreted or what proteins in the blood in circulation in those patients, and those proteins are really like IL-4, IL-13. Those are type 2 genes, type 2 cytokines. All right. And so I think that's cool. You understand what's going on, but how can we take advantage of this mechanistic understanding to potentially make a better drug, right? So even though we know, based on understanding, we know how to make better CAR T cells, but we still cannot test that right away in patients, but we can do that in this leukemic mouse model. We came up with two different strategies to make a better CAR T cells for kind of long-term response. One is you can prime the CAR T cells with the type 2 genes, type 2 cytokines, and co-infuse into the patients, co-administer in the patients. Number two, maybe you can actually kind of prime the cytokines, prime the culture of the cells, that should be right here, and during the manufacturing process. But after manufacturing, you just harvest the CAR T cells. But in this case, you always manufacture much better fitness CAR T cells. They have better chance, better, higher probability to give rise to long-term response, long-term remission patients. So now we test that in the animal model. Okay, without treatment, the mice die within 2 weeks. And no matter use the type 2 low or type 2 high CAR T cells, and the mice always respond. But the type 2 low, type 2 function low CAR T cells, and you can re-challenge the mice, which mimics the relapse. And I think most of the mice cannot respond very well. I think after a month and 3 of the mice already die. But if you use the two different strategies I described here, either you prime, you can co-administer, or you use the kind of interleukin-4 in a manufacturing process to make a better quality CAR T cells. And either strategy can lead to significant increase of long-term remission. In this case, we challenged the mice another time, and actually we did that even one more time to mimic the relapse. And the mice treated in particular with the high-quality CAR T manufactured with the type 2 cytokine in the beginning, give even more striking response. I think after re-challenging the mice multiple times, the mice just automatically reject, and the leukemic cells, you don't even have to give more CAR T cells at all. So that's super exciting. So if we can see that in patients, we pretty much can declare, okay, we can cure every single patient. That's the dream. All right. So pretty much I'm done with my second story, but still a lot of questions remain, right? So in particular, the number 3 here. So majority of the data I show you, that's from the pre-infusion CAR T cells, of manufactured CAR T cells. But what happens when after you infuse the CAR T cell into patients? I know in leukemia it's a little bit difficult to do, because I don't think we can ask the patient to donate some bone marrow and to find out. But in other cases like lymphoma, and it is possible to do kind of post-treatment lymphoma biopsy and see whether or not some CAR T cells still there. If not, and maybe you can still find out how does that change the tumor microenvironment to really understand the biology in much more complete biology story, not just in the drug, but also in the host. So to do that, you need a much better tool to characterize in a spatial context. But I really want to emphasize it's a huge teamwork. I think 4 different institutions and a Penn and a Yale Penn and Cleveland Clinic and my friends working on CAR T cell engineering at EPF Li Tang and at Yale CD Chen. But how to understand better in the spatial in sort of the tumor microenvironment. In recent years, a number of tools have been developed. The space also exploding, and now it's the space exploding, and the space exploding to better understand the spatial biology. And so, just what my lab specific contribute to the field is can be summarized in this slide, this roadmap slide. When Professor Joachim Lamberg published the first paper to use the DNA microarray to capture transcripts from the whole tissue slice you place on top of the DNA microarray. And so many different technologies were reported, but they all follow the same fundamental principles. You take the RNA out of the tissue and they capture on the barcode, the solid surface substrate. But what we came up with kind of total opposite. Instead of capturing, we send the DNA barcodes into the tissue to barcode either transcripts or proteins or many other things. So it turns out it's extremely versatile. So we published a number of papers to show how the technology works, but also further expand this to the spatial epigenomics, which is really my passion to understand the epigenetic mechanism rather than gene expression, rather than protein expression. So recently we have a review article to highlight how the microtechnologies are driving the revolution of the single cell and the spatial multi-omics. Just want to kind of show you, if you get there, what are you going to see? Okay, I really think it's mind-blowing. And this is one of the samples we mapped out is a lymphoma, since we're excited about how we can use CAR T cell to treat lymphoma. And we start with just mapping the lymphoma using our spatial multi-omic sequencing. In particular, our technology allows you to sequence not just gene expression, you can map out the isoform, you can map out even important mutations, there's large small RNAs and tRNAs and nascent RNAs and TCR, BCR, and so on and so forth. And you can further stack that with the epigenome, epitranscriptome, the information extremely rich. And also use the AI machine learning to further integrate the tissue histology image. This is integrated with tissue histology image. You get a truly single cell you can visualize, and every cell you see here, you can ask a question about anything related to RNA, related to epigenome. You are not limited to gene expression. So extremely rich biology now you can explore in ourselves, our human beings. So I think this is another, I think it's the turning point in the whole history of the single cell spatial omics, and where it's about the time to explore the human biology using those truly enabling tools. All right, I'm gonna stop here and just thank all my current former lab members, amazing collaborators, and funding support. And thank you very much for your attention.
Okay, thank you very much. It was fascinating talk. Very nice overview of the history of single cell proteomics and then transcriptomics, as well as how these multi-omics approaches could help patients. Very, very interesting. I had a question about the pathways you mentioned. So the interleukin-4, interleukin-13, this type 2. So priming cells seems to have an effect, a positive effect on the CAR T cells. Do you think this is like something instantaneous where the cells act and then over a short amount of time, or is it a sustained effect, would you say?
Uh, so this is a good question. I think how long gonna sustain, right? So this we did look at epigenetics. At least that's not just a kind of trends in the protein signaling, and you do see the difference in accessibility in chromatin. But I would like to say the T cells always very plastic, but as long as you have the right drug and a good drug to start with, you have much better chance to maintain long-term response. And the cells know what they should do, but if the drug you give in the beginning is not well balanced or it's not the best drug, and the interaction with the host is somewhat compromised, and and then the CAR T cells, no matter how they want to maintain their full fitness, the homeostatic state, they just cannot because this is a game. It's a real game between the drug and the tumors, or between the T cells, CAR T cells, and the tumor. If the CAR T cells are weak, okay, the tumor cells gonna hammer them down, and they cannot exercise their full power. Yeah. So we actually figure out this great question. We actually figure out the mechanism how the kind of either wake or the hyperactive CAR T cells. Either one is not good. All right. You need a perfect homeostatic or perfect fitness CAR T cells. If they're overreactive, turns out that's bad because they can become exhausted very quickly. If they're weak, okay, they don't have good metabolic fitness, and they keep bothering those tumor cells post-infusion, but they are unable to completely eradicate the tumor cells. But they stay alive. The CAR T cells stay alive in vivo. They are just kind of keep harassing the tumor cells. But one day the tumor cells are okay. I got very annoyed. I'm gonna downregulate my CD19 and express something else, and then you don't bother me again. You don't bother me anymore. So I think that eventually we figure out very different mechanisms for the patient to develop relapse, but total unexpected. The mechanism actually can trace back to the T cells you give to the patients in the beginning. Yeah.
Okay, nice. I just have a comment for the attendees. I see some people are raising their hands. If you could type in your questions in the Q&A, then we can ask them to the speaker. Thank you. Okay, so we have some questions from the audience, and one of the questions is, how do you do the power analysis for the sample size, given the heterogeneous nature of the clinical samples?
Yeah, we always got those questions. So I really think in the very beginning, I think the 5 versus 5, right? 5 relapse versus 5 CR. Even though I still don't know how to do that power analysis, but my guess is, oh, that maybe that's underpower. But even though it's underpower, we already discovered the same thing. So now the 82 patients, I don't have question about the power. So maybe if you want to claim a little bit further, so the subtle difference between the relapse within a year within 2 years, we're under power. But if you group all the patients who develop relapse versus the patients who are long flyer, we pretty much have like half of those, half versus half. Yeah, that's what I said in the beginning. The relapse rate about like a 50%. So in that case, we have almost like a 40 long flyers, 40 long remission versus 40 relapse, and yeah, that's either statistically significant. Yeah.
Okay. Another question is, for the long-term remission patients, do we see a shift from type 1 to type 2 phenotype in the CAR T cells that persist in the patients?
Post-infusion, that's what I mentioned, but no one knows. It's a no way or very difficult to know because I think the first couple of weeks post-infusion, the CAR expansion occur within the first 1 or 2 weeks, right? That time, a lot of CAR T cells in circulation. But once the patient respond, and after a couple of months, and it's so hard to detect the CAR T cells in circulation after a few years, and almost you don't detect those. Even the patients are doing well, they stay in remission. You don't really detect a lot of them. And so I don't think you need to kind of shift them from type 1 to type 2. Even our kind of pre-manufacturing product, I don't think we were shifting the type 1 to type 2. We just have a good mix of different subsets there. As long as they have, it's very like a kind of old Asian philosophy. You need the yin and the yang. You need the right balance. If you give pure drug, right? So it's okay. The type 1 gamma interferon and the granzyme B producing CAR T cells are the ones that are going to kill tumor cells. It's going to isolate those cells, and the infusion patient, no, they're not going to work. You need a mixture of all different subsets of CAR T cells to make the best drug. Yeah. If you somehow kind of re-engineer, turn all type 1 cells into type 2, that's not good either.
All right, so we will have a live question from one of
Okay.
the attendees.
Okay, can you hear me, Professor?
Yeah.
Hello. Okay. So my question is that the tumor microenvironment is a highly complex system. So it's like a network. And when you put, for example, CAR T cells into this environment to tackle with the tumor cells, you may find that you block one pathway, but there are alternative pathways to amend this process, and maybe there are many new mechanisms to further support the proliferation or invasion of tumor cells. So how do we deal with this?
But as I said, we have no idea at all at this moment. That's why the last couple of slides read about the future study to look into the mechanism in the host in the tumor microenvironment. We haven't done anything like that. I think all the single cell sequencing data from infusion product, but it's important. I think in 2 weeks. Number one, for pharmaceutical industry, the highest priority is they want to know what's the best drug, how to manufacture the best drug, right? So basically, okay, the cellular immunotherapy now the drug is the living cell, and so you cannot use the conventional methods like a mass spec to characterize what compound is to tell you whether or not that's the right drug, right? So basically, what we show you is how to know, how to characterize, how to QC your drug, and then how to make a better drug. But the mechanism post infusion, we haven't done that much. But some kind of in vitro cell model study. So we do have kind of leukemic cells and tumor cells, and when we allow them to interact, and we sequence both and see what's going on. So I think CAR T cells are really like a hammer. They are stubborn to some degree, and so after a couple of years of post infusion, I don't know. They are rare. They are memory cells. They hide somewhere in bone marrow. But the first couple of weeks post infusion, they are like a very stubborn. They know what they need to do. They just go straight. They don't care the local microenvironment that much. If you get if you get a good CAR T cells to start with, yeah.
Okay,
but afterwards, they need to evolve. They have to evolve. They have to turn. Most of them gonna disappear, but some of them gonna turn into memory cells, and then they need to find the right home in the body to hide to stay forever to continuously patrol the body to prevent leukemia relapse.
Okay, so I think also this spatial technology is very important, for the research of solid tumors, because you can actually look into this specific microenvironment, and you can look how the cells interact with each other, right? So it's like a direct process.
Yeah, exactly. That's why I was still in the very last slide. That's lymphoma.
Yes.
So yeah, now we're ready to look into CAR T-cell lymphoma immunotherapy. We haven't started, and that will be super exciting project.
Okay. I also want to ask a small question. That is, how do you carry out the how do you just characterize the proteins inside the single cells? Do you use like immunofluorescence, like you use the antibodies to tag these different proteins?
There are surface markers. I think I don't know which one you refer to. I think in the paper we did a single cell Dissect to look at surface markers plus the whole transcriptome by sequencing. But the cytokine data, cytokine secretion data, that was done in a separate experiment, not from the same cell, but the same batch. But we did a antigen-specific stimulation and using the technology I described in the first couple of slides, the single cell secretion device to look at single cell cytokine production. Yeah, that was done in separate experiments, not in the same cell.
Okay. Yeah, for the sake of time, I'm sorry, but we only have 6 minutes left. Maybe I'll give one question, and I appreciate a quick answer. It's a bit complex, but how well are epigenetics and transcriptomics correlated on the single cell level? Yeah.
I think most genes, you see accessibility and gene expression, they are correlated pretty well. But when I say most, that's kind of what you expected, right? But the ones that do not correlate well, I feel that's exciting. That's not a problem. So we haven't done that much sort of the single cell ATAC plus transcriptome. I think a lot of people have more data. We have a lot of data for spatial transcriptome and ATAC, but you can do the same type of analysis. So one specific example I can show you is, so we look at the neuronal cell or the brain so glial cell differentiation, radioglial differentiation towards neuronal cell lineage in mouse brain. And so the radioglia, they're really like stem progenitor cells. When they further differentiate into neuronal lineage, and you look at a transcriptome, it's a very nice monotonic differentiation trajectory. If you use the monocle, shooter time, use UMAP, use Fay, that type of trajectory analysis. But when you look at chromatin accessibility, that's no longer monotonic. Actually, you see bifurcation. So some of the cells, even though at a transcriptional level, they're already committed to neuronal lineage, but they somehow still maintain the epigenetic potential, epigenetic memory for oligodendrocyte cell lineages. If you just look at gene expression, we now know. And so it turns out those cells maybe further down the road upon some challenge, like inflammatory challenge, they have the potential to transdifferentiate into oligodendrocyte lineages, right? So that's kind of the difference between epigenetic and transcriptome, and that difference is not a problem. Okay, if you try to use either the ATAC to create the trajectory versus the gene expression to create the trajectory, so wow, they don't look the same. There's something wrong with that. No, this is right. Actually, you see interesting biology. In this case, there's an epigenetic memory. All right.
All right, thanks a lot. So if anyone has more questions, you can always contact us, and yeah, we can try to answer or contact the speaker for some answers. Thanks a lot, everyone. Thanks a lot to the speaker and to everyone who participated or asked questions. If you like this webinar, please register for the next ones in the series. There's a link over there that was posted in the chat, and please also submit your feedback. And with that, I would like to again thank our speaker, thank our panelists, and everyone who participated. Thanks a lot, and see you next time.
Yeah, thank you, Khalil, and thank you for having me.
Oh, it was a pleasure, really.